Hello guys, welcome to Big Book. Today we are going to discuss on human karyotype. We have already uploaded one presentation about the karyotyping. As we all know the definition that the karyotype is a standardized arrangement of the metaphasic chromosomes according to their size and the centromeric position in the decreasing manner with every detail of the chromosome morphology. Now here in the next slide we will see the samples used to prepare the chromosomal karyotype. That is, if it is a prenatal diagnosis, then the samples will be of the amniotic fluid, fetal blood, chorionic villi sample, umbilical cord, and other embryonal or fetal tissues. So this and all you very well know. So here amniotic fluid is the fluid from the amniotic sac, where amnion fluid will be taken. In that there will be the sample of the cells of the fetus, and also fetal blood means whatever the fetus will be developing, that fetus blood will be taken. And chorionic villi is the junction or the place where there will be the blood vessels of both the mother and the child, or the developing fetus, where it is going to, from where the tissues are going to be taken. That is from the side of whatever the chorionic villi or the tissue has been developed from the fetal side, that tissues will be taken, and according to that the sample, the karyotype will be prepared. Umbilical cord, and other embryonal or fetal tissues, any other tissues can also be taken. Postnatal diagnosis, that is after the birth. Prenatal is before the birth. If you want to do the analysis, that is the prenatal diagnosis. If it is after the birth, that is the postnatal diagnosis will be done. That will be with the help of the peripheral blood, that is from the veins, how we take blood in the syringe. With the help of that we are going to take the lymphocytes and we are going to culture them, and from that we are going to extract the metaphasic chromosomal complement, and then we are going to develop the karyotype. Otherwise from the bone marrow, you know what is bone marrow. Otherwise from the skin cells, it is the easily available outer tissue. Carcinoma samples, if it is a cancer patient, then it is a carcinoma samples, and other tissues, if it is the samples can also be taken from the other tissues. So this is postnatal diagnosis. Now this is the depiction, that is the picture which is showing the flow chart, how we are going to develop a karyotype from the samples. So here they have taken the sample from the venous blood with heparin. Now here then that venous blood with heparin has been added with the phytohemoagglutinin and the culture medium. Now if you are going to consider what is heparin. Heparin is an anticoagulant which will prevent the formation of blood clots, and it is also used to prevent blood clots in veins and also arteries or lungs, etc. It is also used in surgery to reduce the risk of blood clots. So the main purpose or the significance of the heparin is to reduce the blood clotting or to avoid the blood clotting. It is an anticoagulant. Now here if you are going to speak about phytohemoagglutinin, it is a plant protein actually, so especially extracted from the French bean. It is going to clump the blood. So here before one which we discussed is the heparin, and now what we are discussing is about the phytohemoagglutinin. Heparin is the anticoagulant, but the phytohemoagglutinin is it is going to cause the clumping of the blood together. Okay, it is not going to form the different grow clumps, but a single clump it is going to be formed. So because of this significance, it has been used in the culturing of the blood cells, that is the lymphocytes, in a culturing medium. So here the culture will be incubated for 72 hours, that is after with the culture medium and phytohemagglutinin for 72 hours at 37 degrees centigrade. So here during this time the culture is going to take part in many cell divisions, many generations of the cell divisions, and after that, after 72 hours, they are going to add colchicine. Colchicine is one lymphocyte culture cells are exposed to this particular colchicine to arrest cell division, especially at metaphase, to visualize chromosomes. Now here this colchicine is going to arrest the cell division. It is going to neutralize the cell division process, and it is going to stop the cell division process at a particular phase, that is the metaphase, so that the highly condensed chromosomes which are needed for the karyotype can be extracted. Further, this culture which has been already subjected with colchicine to arrest the metaphasic phase of the mitotic cell division in the culture is further again subjected to the hypotonic saline. Hypotonic saline is the saline of the potassium chloride salt. This hypotonic saline is going in this culture will be subjected or treated. Because of this hypotonic saline, the cells are going to absorb this saline and they are going to bulge like a balloon form. Balloon form means, in the sense, if you are going to observe in the microscope, they are going to appear to be as the balloon form, not like a big balloon what we see from our naked eyes. So this is how. Further, this hypotonic saline is after treating with the hypotonic saline, further the culture is subjected to the fixation. Fixation means, you know already that you have gone through the fixing of the slides with the samples of the same way by taking the cultures of different organism cells. It means that we are going to kill the cells. We fix them with a methanol is to acetic, like 2:1 ratio or 3:1 ratio solution, and the cells were disrupted when this disrupted in the sense we are going to spread those sample under a cover slip, and further we are going to observe. But here also the fixation is the same with the help of the methanol is to acetic ratio, and here after fixing we are going to take the solution in a dropper. So here in the dropper we are going to take the solution with a height of a meter from the shoulder. We are going to drop a drop of the sample of the fixed solution. So from a height, a dropper will be placed and a slide will be placed just below to it. Just below to, in the sense, it should be at a far distance, where if you are going to drop a drop of this sample, then that particular drop has to fall on the slide in such a way that it has to spread in the force. Because of this, what will happen, whatever this sample, that is the cultured hypotonic treated cells will be there, which are already discussed that it will be burst in condition, they are going to burst open. As we have already discussed that these cells are already arrested at the metaphasic phase because of the calcicin. The chromosomes which are in the metaphasic phase are going to spread on this slide, and further when we are going to take this slide, and if you are going to stain either with the Giemsa or the trypsin stains, it is Giemsa stain, by digesting with the trypsin. So further we can observe this particular metaphysic complement, chromosomal complement. This is what a particular procedure of the how karyotype is prepared with the postnatal samples in the analysis. So further we will discuss, see this slide. This is how a complement will be observed after dropping the sample on this slide. And if you are going to observe this kind of many group of chromosomes will be present, because a drop will contain many number of the cells. And here this will be taken into consideration, and this particular micrograph will be enlarged, and further now these are going to cut into pieces according to the size and arranged. Now before do that, we have to understand in karyotype what is the chromosome morphology. So this is what chromosome morphology you will study very well in many classes of your colleges or in your tuitions or tutorials. Here you can see there is a metacentric chromosome, there is a submetacentric chromosome, there is a acrocentric chromosome. Metacentric chromosome means the centromere is placed in the center of the chromosome by giving the petite and Q arm the same size. That is, both the arms will be having the same size approximately. And submetacentric center is where the Q arm will be longer compared to the P arm, and sometimes you can observe the telomeres in submetacentric centers also. And further is the acrocentric, that is the Q arm will be very long and the absence of the P arm will be observed. So here there will be satellite bodies, which are there are going to be minute bodies on the messiah where the P arm should be present, but there will be the satellite bodies. This is what the chromosome morphology will be in the metaphasic phases of many organisms. So here, especially in human karyotype, there will be 7 groups depending on length and morphology of the chromosome. So here 22 pairs of homologous chromosomes, which are again called to be as the autosomes, will be present. 1 pair of the sex chromosome in male sex, and in female sex the XX will be observed. Now this is how we are going to arrange the human karyotype. There will be group A and there will be group B, there will be group C, which is the biggest or largest group, there will be group D, and there will be group E, group F, group G, and F. Okay, here we can see that A group has 1, 2 and 3 chromosome, that is 1 to 2, 1 to 3, that is 1, 2 and 3 chromosome. 4 and 5 in the B group, 6, 7, 8, 9, 10, 11, 12 in the C group, and 13, 14, 15 in the D group, and 16, 17, 18 in the E group, and 19 and 20 in the F group, only 2 chromosomes here. 21 and 22 in the G group, only 2 chromosomes. Well guys, you can see in this karyotypic complement which has been already arranged, it is of the Jigsaw staining technique, where you can see that 1, 2 and 3 are in the same size, that is the size of the chromosomes are similar, and approximately all the centromeres are metacentric. Okay, and in the second group you can see 4 and 5 the chromosomes are present. These are little shorter or smaller in size compared to the 1, 2 and 3. So here again the chromosomes are having the centromere submetacentric kind. Coming to this group C, 6, 7, 8, 9, 10, 11, 12. So here you can see the chromosomes has been arranged according to the decreasing order in the size compared to the group A and group B, that is 1, 2 and 3 chromosome of group A and 4 and 5 chromosome of group B. So this is the smaller, and further here you can see 13, 14 and 15 and 16, 17, 18, 19, 20, 21 and 22. 13, 14 and 15 forms the D group. 16, 17 and 18 forms the E group, and this is going to 19 and 20 forms the F group, and 21 and 22 going to form the G group. But X, Y and XX separately they are not going to form any kind of the group, but I will discuss in the next slide what kind of group they are going to belong to. So here this is what the actual description about the grouping within the human karyotype. Now here in this you can see in 7 groups of human chromosomes. Chromosome 1, 2 and 3 are the large metacentric, so they are placed in the group A. In group B, 4 and 5 large submetacentric chromosomes are placed. The centromere is submetacentric. Group C will have X chromosome, that is already when I have spoken 6, 7, 8, 9, 10 and 11, 12. The group and the X chromosome which are medium acrocentric, so the centromere will be little more positioned towards the P arm side, that is the acrocentric kind, so giving the Q arm to be longer, so that is going to be the medium acrocentric. So in this group the X chromosome will be placed, because the size of the X chromosome is going to comparably equal to the 6th, 7th, 8th, 9th, 10th, 11th and 12th chromosome. So here the X chromosome will be placed. Group D will be having 13, 14 and 15 medium acrocentric chromosomes. Group E will be having 16, 17, 18 short submetacentric chromosomes, and group F will be having 19 and 20 short metacentric chromosomes, and group G will be having 21 and 22 and Y short acrocentric chromosomes. Here you can understand that the group Y belongs to the group G, and group chromosome Y belongs to the group G, and the chromosome X belongs to the group C. Now here how we are going to depict the karyotype, how we are going to write the karyotype. So that is we are going to understand. We can write the karyotypes two ways, as autosomes plus allosomes, that is 44 autosomes plus XY normal male, otherwise 44 autosomes plus XX normal female. This also can be written as 44 capital A and capital A plus XY for normal male. In the same way 44 AA plus XX normal female. So this is a one way of writing the karyotype, and the other way of writing the karyotype is total chromosomes and the sex of the individual. Total chromosomes means here the number of the total chromosomes of the individual will be given, that is all the autosomes and the allosomes will be included, that is the complete ploidy nature will be included, that is the 46 or the total chromosomes of the human being, including the sex chromosomes, autosomes and the sex chromosomes. So here, after that, to mention the male, that is the sex of the individual, we will write that the sex chromosomes, that is the 46 are the total chromosomes, and it is of the male, that is the XY. 46 XY, it is the normal male. 46 XX, it is the normal female. I hope people have understood this. Coming to next slide, how to number the aberration karyotype, a little different to the normal karyotype. Now here, if you are going to consider, there is a Down syndrome 21 or trisomy 21, there is Klinefelter syndrome XXY, 47 XXY Klinefelter, 48 XXXY Klinefelter syndrome, there is 49 XXXXY Klinefelter syndrome, which is not mentioned here. There is a metafemale, that is 48 chromosome with 4 X chromosomes. Karyotype syndrome, that is 46 XY 5P minus 46 XX 5P minus. So here we are going to observe one thing, that is in Down syndrome we are mentioning the karyotype as 47XX plus 21 trisomy. Here 47, as we discussed, the whatever the complete complement is present in the in the cell, that will be mentioned, that is the chromosome, total number of chromosomes is mentioned, that is 47, including the autosome and the allosomes. Further, we are mentioning the sex of the individual, either the female or the male. Further, we are mentioning what is the extra X chromosome, or the what is the aberration has caused, that is the plus 21 chromosome, that is the 21st autosome has been the extra chromosome in this Down syndrome, that is the 3 number of 21 chromosomes are present. So the Down syndrome is also called as the trisomy 21. Well guys, in Klinefelter syndrome, here it is mentioned with XXY always, as in Down syndrome whatever the chromosome was extra, as it is the autosome, there the number of the chromosome has been mentioned. As the Klinefelter syndrome is because of the allosome, here the sex chromosome has been mentioned here. So the total number of the chromosomes already mentioned, that is the 47, and the sex of the individuals is mentioned, that is the XXY. Here why it is mentioned XXY, it is because the whatever the total number of the allosomes are present, that can be mentioned with their particular denotation of the X or the Y, so it has been mentioned. So here the X has been the extra chromosome, that is in place of the XY, X has been the extra chromosome. Because of that, the Klinefelter syndrome has formed. So in this, the Klinefelter syndrome causes are observed in the male itself, that is because whatever the number of the chromosomes of X may be present in the individual, but the Y chromosome is going to dominate on this X chromosome, because of the maleness developing genes will be present in the Y chromosome, which are dominant over the whatever the feminine gender developing genes in the X chromosome. So here if it is the 47XXY Klinefelter syndrome, or 48XXXY Klinefelter syndrome, or 49XXXXY Klinefelter syndrome, let it be 2XY Klinefelter syndrome, 3XY Klinefelter syndrome, or 4XY Klinefelter syndrome, the individual will remain the male. So here this is how we are going to mention the karyotype of this particular aberration kind. Further is the 48 4x metafemale. Here it is called as the meta female, or again it is called to be as the super female also, because of the 4x chromosomes. Further, one more is the Karyotype syndrome. Here it is the structural aberration, it is not the numerical aberration. Down syndrome is the numerical aberration. Klinefelter syndrome is also a numerical aberration, because there should be one X chromosome in the male, there are two X chromosomes, so here it is also the numerical aberration. There should be two X chromosomes in the female, but there are four X chromosomes in the meta female, so here it is again the numerical aberration. When you are going to consider the Karyotype syndrome, here it is the structural aberration, where here you are going to see that the upper arm, that is the P arm of the fifth autosomal chromosome, will be having the certain part will be going to have the loss of the genetic material. Because of that, there will be the Karyotype syndrome. So here we are going to mention it to be whatever the total number of the chromosomes present. Because of the only the part of the chromosome has been absent or in the loss form, there will be all 46 chromosomes, so all 46 chromosomes will be mentioned, and further the sex of the individuals will be mentioned, that is the XY, and the fifth chromosome on the P arm, the negative sign will be mentioned, that is the 46 XY 5P minus will be mentioned. Here in the same way, if it is for the female, 46 XX 5P minus will be mentioned. So here this is the slide for the Klinefelter syndrome. So here you can observe that this is the XX, here it is the Y chromosome, 2X and 1Y chromosome. This is the karyotype. Now here don't ask that there are no A, B, C, D, E, F, G, okay, but here to understand the syndromic condition, they have made all the homologous chromosomes arranged together, okay, but the X will come again in the C group and the Y will come in the G group. Further, it is again the previous slide is 2XY, this slide is 4XY. So here you can see 4X chromosomes here, and here you can see one Y chromosome. So 4X Y, this is the one more karyotype. Next is this. In this slide you can observe the you have to observe the Down syndrome. Now here you can see that the 21st chromosome is in the 3 number, that is in the G group. You can see that the Down syndrome is showing the 21st chromosome in 3 number. Here again it is the same 47 XY plus 21, here also 47 XY plus 21. So this is again the G binding, IMSA binding. Further is Karyotype syndrome. Here you can see the P arm has got deleted. Here there is a difference between this karyotypic form and this karyotypic form. Here the single chromosomes has been arranged, here the sister chromatids has been arranged. So there is the difference, no need to get confused between these. So here the depiction is showing that the one of the P arm has got deleted. To whichever the individual this particular P arm is going to get segregated or inherited, that individual is going to suffer from the Karyotype syndrome. So here it is 46 XY. So here it is the XY, X and Y chromosome, minus 5P minus. So here this is the 5p minus. So this is again the Karyotype syndrome. Here you can see. So this is some brief discussion about the human karyotype. So here significance of karyotyping is that one is that we can understand how the structure of the chromosome and the morphology of the chromosome is going to appear. That is the one. Second is we can study the staining of the regions, that is where the heterochromatic region is present, where the euchromatic region is present, depending upon the kinds of the staining we do. Third is we can study the numerical aberrations very well, as we have already studied. And also fourth is we can study the structural aberrations also. For example, we have studied about Karyotype syndrome, where we see the deletion of the chromosome part. We can even see the duplication also in some chromosomes. The duplication of some genetic material is going to take part. Because of that, the homologous, out of the homologous chromosome, one homologous chromosome will be having the length little bigger compared to the other. In the same way, if you are going to see the translocations, that is the one part of the chromosome is get cut and translocated to the other homologous pair of within the chromosome complement during the cell divisions. So that is going to cause the translocation, that is again a structural aberration. In the same manner, inversions we can observe that during the cell division the genetic material is get cut and get inverted within the chromosome. Because of that, the homologous looping is going to form in the different manner. In the same manner, in the translocation also, the looping is going to form in the, that is the homologous pairing is going to form in the different manner. The loops are going to appear, so those are called to be as the translocation loops or inversion loops, so depending upon the structural aberration. So this we can study. And apart from this, we will have a particular follow-up of that individual, whether that individual is genetically healthy or genetically diseased. That is genetically diseased in the sense, if these kind of the aberrations are observed in the karyotype complement, that is the chromosomal complement, then that individual is genetically aberrant. That individual is genetically having the changes. Because of that, that individual is going to either going to suffer by the birth, by the time of the birth itself, or in the later in the development and the growth state. So because of this, we can make sure with the carrying people or the parents who is carrying the fetus, if the fetus is normal, we can advise them to have the child. If the fetus is having the genetically aberrant individual or the fetus, then we can ask those parents to get aborted of that particular fetus. So this is what the significance of the karyotyping. So this sends a brief description about the karyotyping. So thank you guys for your help and being tuning with the big book, and we will come with the new slides for PUC of NCERT syllabus, and for degree also, even for BU syllabus and many more. So we will request you people to spread these presentations and slides to too many of your friends or your classmates or whoever is in the requirement of this kind of the presentations. We request your comments below to the videos. How do you like this video, and if there is any kind of the improvement required, please advise us so we can take forward your advices and develop the video or the presentation. Thank you very much. Please subscribe to the big book and press the bell button.